Hello, this is Pankaj Kumar Sinha. In this video, we will discuss about structure of plastids. So let's start the topic. Structure of plastids. Here indicate the diagram of plastids. So labeling here. Outer membrane, it is inner membrane, okay. Grana, double standard circular DNA, okay. This one is Rubisco, ribosome, thylakoid membrane, and in thylakoid membrane or on the membrane of thylakoid quantosome, this one is Freight channel or Stroma Thylakoid, okay. And matrix or stroma, it is also called a stroma, okay. So be labeled here the parts of plastids: outer membrane, inner membrane, grana, thylakoid membrane arranged in a stack like manner, and this one is Freight channel, connect between one grana to the another grana. This one is Rubisco, and thylakoid membrane, ribosome, quantosome, matrix, okay. So we will discuss here one by one parts of plastids. So first part, first of all, plastids. So plastids double membrane structure, double membranous structure like mitochondria. Here outer membrane. Outer membrane contain more porins protein than inner membrane. That's why outer membrane is more permeable. So here key points of outer membrane is porins protein. Okay, so porins protein is the important point of outer membrane. Here porins protein, porins protein present in outer membranes. That's why it is more permeable. Okay, next one is matrix or stroma. Matrix or stroma. So all parts of the plastids present in stroma like double circular DNA, grana, Rubisco, thylakoid membrane, ribosome, quantosome, Fred channel, stroma thylakoid. So all parts present in matrix or stroma. So let's discuss one by one. So first, grana. What is grana? The answer is grana are arranged in stack like piles of coins called grana. So grana is the arrangement of stack like arrangement. Stack like arrangement means if we take some coins, so 1 coin, 2 coin, 3 coin, 4 coin, just it is arranged in stack like manner, so form grana. Here one thylakoid, second thylakoid, fourth thylakoid, fifth thylakoid. So here form whole structure is grana. Okay. Sorry, thylakoid are arranged in stock like the piles of coins called grana. Okay, next point of grana, H chloroplast contain 40 to 100 granum. Granum is singular and grana is plural. Okay, so next point, in granum, in thylakoid membrane, a space formed that is called lumen. In thylakoid membrane, a space formed is called lumen. Okay, so next one is quantazome. In one granum about in one granum quantazome is the functional unit of photosynthesis. Quantazome is the functional unit of photosynthesis, right? Next, in quantazome, contain about 240 to 250 different photosynthetic pigment. Quantazome contain about 240 to 250 different types of photosynthetic pigment. So here photosynthetic pigment, in which type of photosynthetic pigment present here? Chlorophyll A, chlorophyll B, carotene, xanthophyll, etc. Okay, next point, this one is Fred channel or Stroma Thylakoid. What is Stroma Thylakoid or Fred channel? So simply we see here connection between one grana to another granum. Fred channel is the main function of connect between one granum to the another granum. Okay, so right here Fred channel or stroma lamellae connect between one granum to the another granum. Okay, so we discussed here grana, Fred channel, quantosome. Okay, next in matrix or in stroma. Okay, a important point related to grana is also discussed here. Grana is the site of light reaction due to presence of chlorophyll molecule chlorophyll pigment. Okay, now we will discuss stroma or matrix. So first of all, stroma is the site of dark reaction or Calvin cycle. In a stroma or matrix, here present enzyme Rubisco. Okay, due to presence of Rubisco, it perform dark reactions or Calvin cycle, right? Next in Rubisco is the most abundant on the earth. Okay, so Rubisco, and what is the function of Rubisco? Rubisco perform dark reactions or Calvin cycle. In dark reactions and Calvin cycles, basically formation of carbohydrates molecule. In dark reaction, synthesis of carbohydrates, especially glucose and starch molecule. Rubisco also help in 16% protein synthesis. Okay, so we have discussed stroma or matrix, stroma or matrix here. Enzymes present. Which enzyme? Enzyme is Rubisco, right? Next, next in matrix also present double circular DNA or and ribosome. So DNA in plastids double circular, double standard circular DNA present, which is play a major role in cytoplasmic inheritance, okay. DNA is clear. Next, ribosome. Ribosome also present in matrix part or stroma. In matrix, ribosome present 70S type. Okay, so just a minute. In matrix 70S type of ribosome present. Okay, due to 70S type of ribosome and double standard circular DNA, plastids play a role in semi-autonomous cell organelle, or it is act as a semi-autonomous cell organelle. Due to ribosome and DNA, plastids act as semi-autonomous cell organelles. Okay, semi-autonomous cell organelle. And DNA and ribosome, it also helps in protein synthesis. In this segment, we have discussed some important structure of plastids, and now we will discuss about functions of plastids. Functions of plastids. function of plastids. Okay, so here first function is photosynthesis, chloroplast trap or chlorophyll, especially we can say that chlorophyll molecule trap the light energy. Okay, so here chlorophyll molecule trap the light energy and convert into chemical energy and form carbohydrates in the form of food. Okay, so next functions of plastids. Plastids play a major role in balance the oxygen and carbon dioxide molecule. Okay, third function provide greenery on earth. Next function plastids store vitamin E, K, Rubisco and iron molecule, etc. Okay, so we have discussed here functions of plastids. In first function, perform photosynthesis, balance the oxygen and carbon dioxide molecule. Third function, provide greeneries on earth. Next function, plastid store vitamin E, K, Rubisco and iron molecule. Okay, so in this segment or in this video we have discussed about structure and functions of plastids. So we will discuss next topic in the next video. Okay, thank you.